Hi everyone, my name is Macy, and in this video, I'm going to highlight one of the key research techniques used in the Wolberg Lab at UNC, the washed platelet preparation. This might sound like a behind-the-scenes lab detail, but it's actually foundational to how the lab uncovers the biology of blood clotting. By isolating and washing platelets, the lab creates a clean system to explore how these cells behave, especially how they interact with and regulate clot-stabilizing proteins like FX3A. So, what are platelets and why do we need to wash them? If we back up for a second and explore platelets, we find that platelets are small, disc-shaped cell fragments in our blood. Their job is to stick to damaged blood vessels, clump together, and help stop bleeding. They're like biological first responders, but they don't work alone. Platelets carry and respond to tons of proteins that help form and stabilize clots. Now, when scientists want to study what platelets are doing, say how they release a certain protein or respond to a stimulus, it gets tricky if there are a lot of other things floating around in the sample, such as plasma proteins or red blood cells. So that's why we wash platelets. By separating them from everything else in the blood, researchers can create a controlled environment where they know exactly what's happening and what's being added. This way, they can be sure that any reaction is coming from platelets themselves and not something else. So, how does the Wolberg Lab do this? First, they draw fresh blood from human volunteers or from mice. The blood is treated with a compound to prevent clotting before being spun in a centrifuge. This separates the denser red and white blood cells from the lighter platelet-rich plasma. From there, the platelet-rich layer is carefully collected, then spun again to isolate just the platelets. At this point, they're washed multiple times in a special buffer to remove any remaining plasma proteins. Finally, the result was a test tube full of platelets with no red blood cells, no white blood cells, and no interfering proteins, just platelets and a blank slate. So why does it matter? With washed platelets, the Wolberg Lab can then perform targeted experiments. The diagram on the screen shows all the different things that platelet activation can have on a person. This is what they did in one of their recent studies on FX3A, a protein that helps stabilize clots by forming strong crosslinks in the fibrin mesh. By using washed platelets, they could directly test how much FX3A is released versus how much stays inside the cell after activation. Surprisingly, they found that more than 80% of FX3A stays inside activated platelets, even under a strong stimulation. This is only possible to measure because the system was clean. Without washed platelets, plasma FX3A would have made it impossible to tell where the signal was really coming from. The lab also uses this approach to study signaling pathways, cytoskeletal changes, and protein localization, sometimes even using inhibitors to see what happens when certain steps are blocked. Washing platelets gives them the control needed to ask very specific, focused questions. Another powerful part of the wash platelet approach is that it works with the mouse models too. Scientists often use mice for research because they're genetically and biologically similar to humans, making them good models for studying. The Wolberg Lab often uses mice with specific gene knockouts. By preparing washed platelets from these mice, researchers can compare how normal versus altered platelets respond to the same exact stimulation of FX3A. This is especially useful for studying genetic contributions to clotting disorders and for identifying potential drug targets. So to wrap it all up, washed platelet prep might sound like a routine lab technique, but in the Wolberg Lab, it's a key that unlocks a deeper understanding of how platelets behave, how they store and release clotting factors, and how different signaling pathways influence their function. It's a simple idea with powerful results, and it's helping scientists get clearer answers to complex questions about bleeding, thrombosis, and clot stability. Thank you for watching.